In this clip, we're going to travel into the depths of a bacteria cell using virtual reality. Bacteria are prokaryotic cells that have existed on Earth for over 4 billion years. Here, in this virtual environment, you can see a variety of bacterial shapes and sizes. Bacteria can be found in bacilli, cocci, spirochetes, spirilla, vibrios, or even some other unusual shapes. Two types of appendages are visible on the surface of bacteria. Some bacteria are motile. These bacteria have an appendage known as a flagellum. Flagella can be interior, such that a microscope cannot see, like this spirochete, which is now moving in this virtual environment. Other kinds of flagella are exterior and can be seen either at one pole, two poles, or around the bacteria. Though the flagellum is a dedicated tool for bacterial movement, it's also an essential antigen of motile bacteria. The second bacterial appendage is the pili, which are made of proteins and can be seen in large numbers around the surface of the bacteria. They are short and not flexible and are the bacteria's tool for sticking to host surfaces. Now, let's take a look at the inner parts of a bacterial cell. Here we've hypothetically crossed the bacteria's capsule, cell wall, and cell membrane, and we're now in the cytoplasmic region of a bacterial cell. The cytoplasm is a jelly-like and dynamic environment. It is 75% water, and the cytoplasmic organelles are floating inside it. The most crucial bacterial organelle in the cytoplasm is the nucleus, which is made of approximately 1 to 2 millimeters of tied and knotted cyclic DNA, and is known as the chromatin network, and contains the primary information of each bacterial cell. The following cytoplasmic organelle that contains DNA is the plasmid, which is only present in some bacteria. It is cyclic, and its size is approximately 1/10 to 1/100th of the length of the bacteria's main chromosome. Its information isn't vital to the cell. Genes related to antibiotic resistance, exotoxins, capsule, and so on might be coded on a plasmid. The information that exists on chromatin and the plasmids are eventually transcribed in the form of mRNAs, and each mRNA molecule gets translated into a protein by positioning on several ribosomes called polysomes. Granules are the following organelles in a bacterial cell and are considered food supplies. Their size, number, and components depend on the type of bacteria and the environment in which it's growing. The plasma membrane of a bacterium folds into the cytoplasm in some parts. These parts are called mesosomes and are where the bacterial enzymes for oxidation come together. Chromosomes usually use the main mesosome in the cell as points of attachment to the cell. This point is also where cell division starts.